So, good evening, everyone. Let's begin with the today's session. Can you see my screen, guys?
Yes, ma'am.
Yes, ma'am.
Okay, fine. So, can anyone tell what is NGS?
Next generation sequence.
Okay, so what else you know about this? What is this NGS? So, see, it is the next.
Sequencing of genomic data.
Okay. So it is the next generation sequencing. So in the earlier time, what happened? We were using the Sanger sequencing. So what is the Sanger sequencing?
It's a long run short.
What?
It's a long run, ma'am. It's a type of a long run sequence.
It's basically high throughput sequencing
Okay.
to validate a specific mutation and variant as well.
Okay. Actually, like
It's a
in the older.
method of DNA sequencing where which involves gel electrophoresis.
Okay, so in the older time, we were using the Sanger sequencing. So in this technique, like it was sequencing only a small segment of the DNA. Okay. So it's a time consuming process. So nowadays we are using the next generation sequencing. Why? Because it is the high throughput technique. What is this high throughput technique? Anyone can tell me if you know? So high throughput technique means it is sequencing millions of fragments at one time. That is why people have shifted from Sanger to PCR to NGS nowadays. Because it's very easy, like nowadays you can fragment million of fragments at one time, right? And also it is the faster process, and also the result is very accurate here. Next, but why NGS? Right, the question is that. So there are certain application. So with this NGS, we can find out the novel mutations or any novel organism or any mutations, if you want to know, that you can also see. Second, if you want to see the gene interaction, that also you can study with the help of NGS. And if you want to study the disease association, that also you can do. For example, if you have a two gene, A and B, and if B gene is causing a disease, and if you want to find out whether that A gene is controlling the role of the B or not, that also you can see. If you want to study the DNA, protein binding. So there are a lot of application in the NGS. But we are going to study the application on the basis of RNA. And our application name is reference-based RNA sequencing. So any idea like what is the aim of doing this reference-based?
It helped to compare the normal and mutated sequence,
Normal and
differentiate between the normal and mutated sequence.
okay. What else?
To identify the
Maybe-
differences and the one we are testing on.
Okay. So basically, in this reference-based RNA sequence, we are going to see the profiling of gene expression by identifying the gene or molecular pathway that are differentially expressed between two or more biological condition. So that means what? Like gene expression is the main thing in the RNA sequencing. Here in this application, we can also analyze the continuous change in the cellular transcriptome. What is a cellular transcriptome?
Ma'am, transcriptome is basically all the RNA content inside a cell, like all the rRNAs, mRNAs, tRNAs.
Right.
Say RNAs, messenger
Okay.
RNA.
Yeah. Transcriptome is basically is what the set of all the RNA molecule. That is the tRNA, rRNA, mRNA, non-coding RNA, right? So whenever we are providing some treatment, if you want to see whether these changes it is happening in the tRNA, RNA, mRNA, that changes we can see from this reference based. Okay. And this RNA-seq, it is very like becoming widely used technology because it is highly sensitive, and also from this application, we are going to see the gene expression. Okay. And it reveal the, or you can say the presence and quantity of RNA in sample.
Hello.
Yeah.
Excuse ma'am. Ma'am, can you please explain cellular transcriptome once again, please?
Okay. So transcriptome is what? The set of all the RNA. That means the tRNA, rRNA, mRNA, or maybe the non-coding RNA. And if I am providing some treatment, okay, and if I want to see the changes that is happening in this RNAs, that you can see with the help of this application. Okay.
Okay, ma'am. Thanks.
And also if you want to see the presence or the quantity or how much quantity like of the RNA that is present in the cell, that we can also see from this. And it is quantifying the differential gene expression. That is very important. What is this differential gene expression?
Upregulation and downregulation.
What? Can you speak louder?
The genes which are upregulated and downregulated.
Yes,
Expression.
ma'am. Yeah. So in the differential gene expression, we are going to compare the different samples. Okay, maybe the control and the treated. And if we want to see like which sample is showing up regulation or the down regulation, that is what the differential gene expression. So in order to get the differential gene expression, at least you require two samples. So in the universally, like we are having the control and the treated sample. So what is this control? That is the normal, healthy, or maybe not diseased. Whereas in the treated is the abnormal and the disease one. So there are different biological analysis plan. So it is based on control versus treated, treated versus control, and it can be the control 1 versus control 2, or maybe treated 1 or versus treated 2. So can anyone tell me, like in this plan, control versus treated, what we are going to do?
Ma'am, we will compare the expression with the control and the treated. Either it's one-fold higher or lower, so we can easily differentiate through DEGs.
So here, like if we are choosing a control versus treated plan, that means we are checking the expression of control with respect to treated. Now, can anyone tell me what we are going to do here in the treated versus control? Imran?
Yes, ma'am. This is just the opposite. I mean, we can compare the treated sample with the control. I think so.
Yeah, so here we are going to check the expression of treated with respect to control. Whereas in this control 1 versus control 2, that means if you want to compare like the brain cell of yours and mine in a same condition, that time we are taking this condition, control 1 versus control 2. Whereas in this T1 versus T2, if any sample, like I'm providing any drug treatment, and if you want to check the two treated sample, that can also be done with the help of treated 1 versus treated 2. But it's up to you which plan you are choosing for this reference based. Is totally on you. Okay.
Ma'am, in
Next,
treatment,
yeah.
excuse me, ma'am, in treatment 1 versus treatment 2, it may vary in dosage as well,
Yeah.
na?
Yeah. It
The
can be.
amount of dosage to be provided.
Yes, ma'am. Yes, ma'am.
Okay.
Now, can anyone tell me what are the 3 common steps in the NGS?
Collecting the raw data.
Okay.
And first is extracting. Getting the genomic data from
Okay. So see, first step is the library preparation, second is the sequencing, and last is the data analysis that bioinformatic people do. Now, let me explain you with the help of diagram. Please mute yourself, guys. So see, I told you at least 2 sample we should have, right? So for example, I'm taking the control. So this is my control. And this is the treated one. So as this application is based on the RNA, so from this control and the treated sample, what we are going to isolate? DNA or RNA?
RNA.
Yeah, right. RNA. So after isolating the RNA, we are just converting this RNA to cDNA. So any idea why we are converting this to cDNA? Why not we are directly taking this RNA?
Because of coding.
What?
Coding region.
Ma'am, RNA is unstable.
Yes.
mRNA.
Yes, yes.
mRNA.
So see, one reason is that the RNA is less stable to DNA. If I'm providing a high temperature to this RNA, it can be degraded easily. So I don't want to degrade my product, right? So that is why the first reason is that because of the stability of the RNA, we are not taking this RNA. Second reason is that in the RNA there is uracil. So uracil cannot be read by any of the NGS sequencing platform. So that is why we are converting this RNA to cDNA with the help of reverse transcriptase enzyme. So after we have the cDNA, we are making this into the small fragments. So any idea why we are creating the fragment of the cDNA now?
Ma'am, please repeat what you're saying. In RNA has uracil, so
See, ma'am, first reason is RNA is less stable. And second reason is that in the RNA, there is uracil. So none of the platform machine can read this uracil. That is why we are converting this. Okay.
okay, ma'am.
Now, next step is to make the fragments. Why? If I take this complete cDNA into the machine won't run because none of the platform can read the entire genome. That is why we are converting the cDNA to the fragments. Clear? After making the fragments, next step is the adapter ligation.
Ma'am, please tell me how we do these fragments.
What?
I
So,
mean, how
see, there are a lot of enzymes, sir. So we are not going to discuss this wet part here. This is a long procedure, sir, here in the fragments. Okay.
okay.
That we are not going to deal with this. This is the wet lab part. But yeah, if time allow, I will tell you how we are creating this fragment, sir. Okay.
Yes, ma'am.
Yeah. So from this sample till the adapter ligation is my first step, that is the library preparation. That is wet lab people do. And after this adapter ligated fragments, what we are doing, we are just sending to the sequencing machine. So second step is what? A sequencing. And in the sequencing, we are getting this raw data in the form of reads. Reads are nothing, these are just small fragments of the DNA. And after sequencing, this is our main file, raw data. And this raw data, it is in the form of FASTQ file. And after this raw data, we are performing the analysis. That this is just a basic step that we do in the NGS. Clear? All the
Ma'am,
steps?
I have a question.
Yeah.
Why we do this adapter ligations? I mean, what is the purpose in fragments?
Okay, so before we discuss this, can you tell me what is this adapter?
No, I don't know. That's why I'm asking.
See, adapters are what? These are short oligonucleotide which are universally labeled and universally in nature. So it is basically what it is doing. It is stretching the, like if you are having only one fragment, sir, okay, this one,
Okay.
without the adapter. And if I'm running the sequencing for this fragment, there may be a chances it can be degraded, right? And also the result which we are getting for this, you might not get appropriate, correct? And why we are
Yes.
adding this adapter and the primer, just to create the polymerization for this, right? So we are creating a millions of fragments so that we cannot lose the data, sir.
Okay.
Okay.
Yes, ma'am.
So this is just a basic step of the NGS. But next is what we are going to do, this reference based. So this is my raw data. This is my control 1 sample. Treated 1 and treated 2. So why I'm taking the replicates of control and treated? Any idea, guys?
To improve the accuracy of the samples.
Any idea? So before we discuss this raw data, so in this reference based RNA sequencing, what happened? The reference genome in database for our sample. The GTF file where all the annotations, it is there. Now this is the control sample and the treated sample. So for example, in the control, we are having only 5 reads. Here in this 10,
Ma'am, your voice is
Ma'am, your voice is not clear.
Hello? Hello? I think ma'am left maybe due to network issue.
Okay, let's wait.
Hello?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Can you see my screen?
Yes, ma'am.
Yes, ma'am. Now it's visible.
Okay. So I have taken the replicates of the control and the treated. So can anyone tell me why I am taking the replicates?
To maintain the integrity of the data.
What?
To maintain the integrity of the data.
Okay. Actually, what happened? See, if I take only one sample for the control and one sample for the treated, there may be a chances like RNA can be degraded in the control one sample, right? So if it is degraded, we are losing the data, right? So at least minimum 2 samples should be there for the replicate because we don't want to lose any data. So in the raw data, after getting that, we are mapping on the reference genome. So for example, this data has been taken for human. So I will download the reference genome of the human only. And for this genome, we already know where the gene 1, gene 2 genes are present, right?
Yes.
So for example, this is the G1 here.
Hello? Hello?
Yes,
Ma'am, your
ma'am.
voice is stuck. Your voice is stuck.
Hello.
Yes, ma'am.
Now you can see the 5 reads are from the control 1 and the control 2. There are 10 reads, right? Now can you tell me like which sample is showing higher expression here?
Control 2.
Control 2, right? Why? Because 10 reads are mapping on this reference genome. So like control 2 is showing 5 times expression than the control 1. Whereas treated 1, it is showing no expression, right? Clear?
Yes, ma'am. Yes, ma'am.
Ma'am, please explain one again about human reference genome.
Okay. So now I will tell you the workflow of this reference based data. So this is my raw data. After this raw data, we are going to perform one tool that is the fast QC. So fast QC tool is what? It is checking the quality of your raw data. So it can be good or bad, right? If
Yes,
it
ma'am.
is good data, it can be fine. If it is bad, bad means what? For the bad,
Poor
there
quality.
is Yeah.
The sequencing is not proper or the adapter molecules are not well ligated. So the
Quality of the
sequencing is
data
not.
is
Okay. Bad means if you see adapters or primers are still there in the sequences, that means this data is the bad. Why? Because we are not going to perform the data on the adapters and the primers, right? We want to study on the actual sequence. So for the bad data, we are going to run another tool, that is the kata depth. So kata depth is what? It is the one of the trimming tool that will remove the adapters. Now, after removing the adapter with the help of kata depth, we are going to check it again whether the quality of data is good or not, right? So again, run the fast QC, and after running the fast QC, you will get a good data. Clear till here?
Yes, ma'am.
Now, after this good data, we are going to perform the mapping tool with the help of HISAT2. HISAT2 is the mapping tool. What is mapping? So here what happened? Yeah, tell me.
Like marking the location of the expressed DNA on the human reference genome.
For example, the data has been taken from human. If the data is taken for human, so make sure your reference should be of human, not for other organism. Okay. Now for this reference genome, we already know all the annotation. That means what is the genes name, and the location of those genes. We already know for this reference, right? With this HISAT2, we are going to map the reads data from the control and the treated over here on the reference genome. So it will map wherever they will find the similarity. Clear?
Ma'am,
Clear,
again,
guys?
please repeat, ma'am.
What?
yes, ma'am. समझ आ गया.
Okay. Now we have mapped from this control and the treated, right? Now further we want to know now how many reads are coming from the control 1 and the control 2, right? So for that, we are going to run another tool, that is the cufflinks. Cufflinks will give you the count of the reads. So in the HISAT2, the reads will be mapped. And if you want to know from this control, 5 reads are coming, 10 reads are coming, that count, that number you will get from this cufflinks. Clear? After this cufflinks, we are going to perform another tool, that is the cuff merge. So cuff merge is what? Can you tell me if you ever heard about this cuff merge tool? What is this cuff merge, guys?
It mean like the sequence which are resemblance with each other, they are merged together. Like
Okay, actually what happened in the cuff merge, you are getting the transcript files, ma'am. So for each file, you will get a transcript file. For example, for the control 1, you are getting, okay, treated 1. Like I will just write here transcript 1 file, transcript 2. So total 4 will be there because 4 samples are there, right? So from each sample, you will get a transcript file. Then these transcript file, okay, we are going to merge together with the help of cuff merge. Why? Because we are going to perform the differential gene expression with the help of cuff diff. So if you are not merging this transcript file, you cannot compare, right? That is why all these 4 transcript files we are merging with the help of cuff merge, and after merging you will get only one transcript file, one huge transcript file, okay? And for this one transcript file, we are going to perform the cuff diff, where we are going to see the differential gene expression. Clear? So at the end, you are getting the differential gene expression file. This file is very important for the analysis. And after this differential gene expression file, we are going to do some post-analysis step. That is, if you want to see significant genes, that will see. Because if you download the entire genome, okay, if we are mapping, there may be a chances some of the genes are not showing less expression, some are showing no expression, and some are showing higher expression, right? So which genes those are showing expression, that is the significant. And out of those significant genes, we are going to see the upper regulated genes. Then we'll see the down regulated genes. And we'll see the pathways. Functional enrichment we will see. Network analysis will do. So all these steps are what the post analysis steps. Clear? All the pipeline?
Yes, ma'am.
Everyone? Any doubt in any steps?
Ma'am, why we are doing cuff merge?
Which one?
Cuff merge.
Cuff merge. See, ma'am,
Yes.
if you are getting a separate file for the transcript, so can you compare your data easily?
Sorry,
No.
ma'am, I don't get you.
See, if you're getting transcript files, right?
For
And
each sample, right?
yeah, for each sample, you are getting 4 different transcript files, correct? And if
Yes.
I ask you to compare your data, so how will you compare your data? You cannot do that, right? So what
Yes,
I
ma'am.
will do, all these transcript file, I will merge together. It will combine together and it will create one file.
For control and treated also, it create one file.
Yes,
Then how
ma'am.
will we differentiate? Oh, for that only we using cuff diff.
Yeah, for the cuff diff, ma'am, we are going to choose the analysis plan there. Whether you want to see the expression of control with respect to treated or treated with respect to control, that analysis plan we have to mention in the cuff diff there. Okay?
I'm sorry, ma'am. If in cuff merge we are getting transcript data of control and treated for all samples.
Yes.
Then in cuff diff, how will we differentiate the control and treat? These are the control
No,
genes
ma'am,
and
it
these
is
are treated.
already labeled, ma'am. It is not merging all the data. So see, control section will be listed differently. Okay, and for the treated, it will be listed differently.
Okay, okay.
Yeah, when you will see the output, you will come to know what we are doing. But for now,
Okay.
like the basic step, is it clear what we are doing in this pipeline? Because in the coming next 10 days, we are going to perform these tools. Clear? Any doubt?
Hi,
Ma'am,
may I ask you something?
Yeah.
Okay, so I have a questions about the it's two-bit question. So may I ask about something read length? in DNA sequencing, basically we have some for short read sequencing, we have some 150 usually read length. So is there anything like here for RNA sequencing, the read length, either 150 or more or shorter? Is there anything something here? Do you get my point?
Yeah.
Okay,
No,
all right.
sir. Like, we are not mentioning here the read length, sir, in this.
I mean, so when we do the sequencing of the RNA,
Yeah.
so what would be the read length of that? Means
Usually,
what
like,
you align with the reference genome. So there should be some read length or something like that.
yeah,
Of
like
different
usually
reads
people
or
are
maybe
using the 50 base pair, sir.
50 base pair. All right. So I got it. It makes sense. Thank you.
Yes. Okay. Any doubt further, guys, in this pipeline?
Ma'am,
What?
initially,
Hello?
Yeah, Dhyuti.
yeah. So
Hello?
initially, when you were talking about the raw data, you said that it was in the GTF file, right? So for the rest,
Will be in the form of fast queue, ma'am.
okay, then what was the GTF file for?
GTF file means what? All the annotations will be there. Okay, so that we
Okay,
will see, ma'am, like when we will map this HISA2 tool. Okay, here we are
okay,
using the GTF file. We'll tell you
all right.
further.
Yeah, okay,
Okay.
okay.
Don't worry.
Hello? In the pipeline
Yeah.
that you gave,
What?
I realized that because different number in the expression and the length, you didn't talk about normalization.
Yeah, sir. That part we'll discuss like later on, because see, in this in today's session we are just having a brief introduction on this pipeline. So what you're talking
Yes.
about, the normalization, that will be covered in the cuffling, sir. So whole
Okay.
1 hour session will be there for each tool, sir.
Okay.
Okay, and we'll discuss the all the output and what are the input. Clear?
Okay.
Yeah. So that normalization part will be covered here in the cuffling, sir. Don't worry with that.
Okay.
Yeah.
Ma'am, one more doubt. If our reads are paired and reads, then do we align straightly, or else we have to merge those reads?
Can you speak it again, ma'am?
Ma'am,
Uh-huh.
can you hear me?
Yeah, actually your voice
If,
is see, I will just give you 10 minutes after this session. Let me just complete this part. Okay, then we'll have queries.
okay.
Okay, yeah. Now, next thing is the replicates. What is this replicate, guys?
Small fragments of RNA and DNA.
So replicates means to check the variability in the sample. So there are two type of replicates. First is the technical. What is this technical, guys?
Ma'am, technical replicates mean if we have a one sample, we will make a 3 replications from that one sample. This is a technical replicate.
Yes, sir. It means same experiment. We are performing on sample again and again. For example, you are having a leaf, and in the leaf, you are having a lot of cells. And what I will do, I will just isolate this cell. This particular cell. And then I am doing the experiment multiple times on this one cell. After that, what we are getting? The copies, right? Correct? So you're getting copy of this one cell. That is what the technical replicate. Then further we are seeing whether it is showing the high, low expression. It's up to you, like what type of data you are dealing with that. So this is just the technical replicate. Clear? If we are performing the same experiment multiple times on one cell, you are getting the replicates of that cell. Clear?
Yes, ma'am.
What is this biological, Imran?
Ma'am, biological means
Well understood.
What is this biological?
in which we have a replication, but from two different samples or at two different situations.
Yeah. So biological replicates is what we are taking the different biological sample within the same population. For example, you are having a 2 hibiscus plant. Okay. And from this like plant, we are taking a leaf. And again, in this leaf, you are having a cells. And for this, again, I will isolate one cell from this 2 different leaf. And then again, I'm going to perform what? The experiment multiple time on these cells. And again, what I will get? The multiple copies, correct? That is what the biological replicate. Clear? What is the difference between the biological and the technical? Any doubt?
Well,
So
I
what
think
is depth,
it's
guys? What?
it's well understood. But my question is, which one must be better than which one is better? Which one will have a better data for analysis? Biological replicate or technical?
Sir,
Which one will be more representative
No,
for generalization?
Sir, it depend on you only. Like it's not about the like which replicates you want to choose. It's depend on the experiment. What kind of experiment you want to perform? See, here also, if I'm taking the control versus treated plan, the result will be different here in the control versus treated and also in the treated versus control. It won't be same, sir.
Okay.
But it depends,
Yeah,
like
yes,
what type of expression or what type of result you want to get it.
Okay. Okay. I'll
Yeah.
get it. Okay.
Now, can anyone tell me what is the depth?
Should be
Have you
the expression.
what? Expression?
Yes, I think it means an expression, how express a gene could be.
No.
Like, okay.
Depth means how many times the base is read by the sequencing machine. That is what the depth. If the sequence, in the sequence there is a adenine, okay, and the sequencing machine, it is reading what 5 times the adenine, that time the depth would be the 5x for the adenine. Clear? Clear? What is this 5x, guys?
Depth of adenine base.
For example, this is my reference, okay, and I'm aligning the reads on this reference genome. Okay, on this reference there is a adenine here, and these are my aligned reads. And sequencing machine is reading, okay, from this 10th position maybe. And what we saw on this 10th position, okay, there is adenine. So how many times it is reading? So it is reading, you can see here, 1. So one time it is reading like the, oh wait, one time it is reading A here, from here, second, 3, 4, 5, 6 and 7, right? So that is what is 7x depth here. Clear? If it is reading 10 times the base, that means the 10x depth is there. Clear? What is depth? Clear?
Yes.
Next is the coverage. Coverage means how many reads are able to cover the reference genome. That is what the coverage. So for example, this is my reference genome. And I'm aligning the reads on this reference genome. And what I saw, the reads have aligned this many. So you can see 50% of data, it is covered right under your reference genome. Correct?
Ma'am, please explain this once again, please.
Yeah. Coverage means how many reads are able to cover the reference genome. So here you can see this is my reference genome. And for any particular organism, I'm going to align this read on this reference genome, right? So what I saw, this reference genome have covered this much, 50% of data, right? But in real time, what I will do, I want to cover my reference genome entirely, correct? So that time, what I will do, I am going to increase the depth. If I making the depth, that is the 10, maybe 10x. So what happened? Next reads will cover the reference genome. And now it will give me what? The 100%, correct? Right? Clear? So that means while increasing the depth, coverage is also getting increased. Clear? What is the depth and the coverage?
Yes, ma'am, little
Yes.
bit. But can we get the practice session of this particular,
Which one?
ma'am? This one coverage.
Okay. So see, like just for now, like you should know what is depth and the coverage, because these words will be seen in the output results. Okay. And Fred quality. So that time I can't discuss like this basic terms. That is why I'm discussing this coverage depth. Okay. But just for now, like we are just saying how many reads are able to cover the reference genome. That is what the coverage is. Okay.
Ma'am,
Okay, ma'am.
hello, ma'am.
Yeah, yeah,
Please,
yeah.
could you give us a real example for this? Because I feel that I can't catch the idea of the coverage.
Okay, okay. So maybe when we will just generate the result, that time we can see what is this basic terms, because that time only we can see the examples, ma'am. But
Okay,
for
thank
now, you
you.
can just browse it little bit in the Google or that's it.
Okay,
Okay.
thank you.
Yeah. What is this Fred quality score, guys? Any idea about this Fred quality score?
Quality of the bases, that is ATGC.
Yeah. So it is assigned to each and every base which is sequenced by the machine. So every base which is sequenced by the machine has some quality score which is telling you about the accuracy, that how accurate the base was called by the machine. So for the Fred quality score, the cutoff is greater than equal to 30. So why we are taking this cutoff greater than equal to 30? So we can see here on this first column, this is the Fred quality score. When the Fred quality score is 10, that time the probability of the base call, you can see 10 time the base was called and 1 time the base was incorrect. And the base call accuracy is 90% there. And when the Fred quality score is 20, that time the base was called 100 times, and out of 100 times, 1 time the base was incorrect. And then base call accuracy is 99%. And when you see thread 30 is the Fred quality score, 1000 times the base was called and 1 time the base call incorrect. Here you can see 99.9. So why we are taking this 30 as the Fred quality score, or maybe it is more than that? Because if we increase the Fred quality score, you can see the base call accuracy is just the 99.99, right? None of it is giving what the 100% base call accuracy. So that is why to get the good quality of your data, your Fred quality should be greater than equal to 30. Clear why we are taking this 30? Any doubt here?
Yes, ma'am, clear.
Okay. So we'll end here and we'll discuss the raw data downloading part in the tomorrow session. And we
Ma'am,
just, yeah, yeah, yeah.
ma'am, the explanation of Fred quality score is like more accurate when we getting it approximate 30 or it is more than 30. So that means the score is good.
Yes, ma'am. It should be greater than equal to 30, ma'am. Cut off.
Okay, ma'am. Thanks.
Okay. Is it clear why we are taking this as the cutoff? Any doubt? So I will share one data with you and just make sure it is downloaded before the session. Because like in tomorrow's session, we are going to proceed with the fast QC. Clear?
Yes, ma'am.
So we'll discuss one thing more, that is the BAM and the SAM file, and then we'll end. So these are different format of your file, that is the BAM and the SAM, which is containing the complete mapping information. This BAM file is what? The binary alignment map. That is what the computer readable file. Whereas the SAM file is what? The sequence alignment file. This is the human readable file. So see, like in the coming session, like we will see what is this binary file, why we are creating this binary file, because tool cannot read this SAM file because this file it is containing the ATGC. Okay. So that
Ma'am, your voice is not clear. Hello? Ma'am, your voice is stuck.
any doubt, guys? Any doubt?
Ma'am, your voice is not.
What?
आपकी आवाज नहीं आ रही थी मैम.
No,
Ma'am,
no,
can you
no. I'm
speak? Your voice is stuck. When you
asking.
write down sequence alignment map, human readable file. So after that, we just don't hear you, ma'am. We
Okay,
don't hear you.
okay. So I told you like these BAM and SAM files are the different format files and it is containing only the mapping information. So these BAM is the binary file which tool need. But if you want to read that binary file, you cannot read that, right? Because it will be in the 0, 1, maybe that number. That only computer can read. But if you want to read it by your eyes, that time you have to convert this BAM to SAM in order to read this ATGC format. And if you want to convert this BAM to SAM, SAM to BAM, that you can do with the help of SAM tools. Clear?
Share a screen, please.
Hello? Any doubt, guys?
Yes, ma'am, it is clear. Thank you.
Okay, so we'll end here and we'll meet tomorrow at 8 p.m. guys. And I will just give you one data. Just make sure it is downloaded in your laptop because it will take time.
Okay.
Yeah. Okay. Then bye bye.
Oh, are you sending the link on WhatsApp?
Yeah. I will just share like maybe on the WhatsApp group, like the data will be shared and you can just download that, and then I will tell you in the tomorrow session from where the data has been taken. Okay?
Okay. Okay.
Yeah.
All right. Thank you.
Thank you. Ma'am.
Okay. Okay, guys.
One more question.
Yeah.
Is there any configuration we have? Do we have to install Ubuntu or RNASeq?
Yes, ma'am. Because in the tomorrow session, we are going to perform the quality check by the fast QC tool. So make sure everyone is having the fast QC tool by tomorrow. Because everything will be run on the terminal, guys.
Okay.
Yeah.
Hello, ma'am.
Yeah.
Hello. In the
Hello.
library. Hello, ma'am. Hello.
Yes,
Hello.
ma'am. I can hear you.
In the library preparation, you told like we are adding the answers and we are making the sequence fragments. That is in the wet lab. That means we are cutting the sequences with the restriction enzymes. Is that correct, or is there any?
Ma'am, your voice is breaking. Can you be little louder, or maybe you can just send your message on the chat box?
Yeah, sure.
Yeah.